Right, so we are going to continue our journey in learning about how stuff crosses the membrane and transport into and out of the cells. But before we can do that, we need to learn a few terms because it is important that cells can transport things into and out of themselves because that helps them maintain the balance of both solutes and water. And I wanted to go over a few terms first, which are hopefully going to be familiar from chemistry. So a solute is anything that is dissolved in a liquid. The liquid that it's dissolved in is a solvent. And when you mix solute plus solvent, that makes a solution. Now, most of the time, the solutions we're dealing with in biology are aqueous solutions, which means that our solvent is going to be water. And just in case you forgot, water is called the universal solvent for a reason. It dissolves a lot of different types of substances. So when you have things dissolved in a liquid, you can measure how much solute is dissolved in that solvent, and that tells you the concentration. The more concentrated something is, or the higher the concentration, the more solute you have dissolved in a certain amount of solvent. When you have differences in concentration across a distance, that's called a concentration gradient, like what you see in the diagram here. On the left-hand side, there are more of those molecules. On the right-hand side, over here, there are less of those molecules. So a difference in concentration across a distance causes a concentration gradient. And when there's a concentration gradient, molecules are going to naturally move from where there's a high concentration to where there's a low concentration. So what you're seeing in this diagram is molecules would move from the left to the right without any input of energy. Since cells are selectively permeable, that allows them to form these concentration gradients of different solutes across a membrane. And if you're wondering why you would want to do that, that is important for the function of neurons, something which we unfortunately no longer talk about in AP biology, but it's going to become very important when we talk about cellular respiration and photosynthesis. So passive transport is hopefully familiar from your freshman year, and that's usually moving molecules from a high concentration to a low concentration without energy. So that is no ATP. Active transport, on the other hand, does use ATP, and that's because usually it's moving things from where there's a low concentration to where there's a high concentration. So that takes energy from the cell. Then, as far as active transport goes, we'll talk in more detail about active and passive transport, but I wanted to move on to bulk transport, which is moving large substances or large amounts of something into and out of the cell. There are two types of this bulk transport, and they both require energy. That is endocytosis and exocytosis. So the name endo means inside, and that means that you're moving stuff into the cell, like what you have in these three diagrams here. There are three different types of endocytosis. You're not required to learn about those for the AP Bio test. And then exocytosis means outside. So you're moving stuff to the outside of the cell. And there's really only one type of exocytosis. So if we look at endocytosis in more detail, it allows the cell to engulf materials. That means to surround it and take in. And there are two different types shown here. The one on the left is a specific type called pinocytosis, literally means cell drinking. And that is the cell taking in a liquid from the surrounding solution. So it's usually to take in some solute. The other type of endocytosis shown here is phagocytosis, which is how some single-celled organisms will eat. Usually they'll take that vesicle that forms when they take in whatever molecule, they'll join it with the lysosome. The lysosome helps digest it, and then the cell can extract nutrients from that new vesicle that's made. Keep in mind that this forms vesicles inside the cell. So those bits of membrane that are pinching off with whatever material you've brought in, those are vesicles. Exocytosis is the opposite of endocytosis. A lot of times it's used when a cell is secreting something. That means it has produced it and released it. So this is going to take a vesicle that already exists inside the cell. It's going to move towards the cell membrane, fuse with it, join with it, and then release whatever molecules or whatever particles are inside of it to the outside of the cell. And that process does use ATP. I'm sorry for all the slash marks. My tablet is being a pain. Well, that's it for this lesson. Next lesson should hopefully be coming in a day or two.